Hi, I'm Doran Goldman, and this is my project, Propagule Size's Context-Dependent Effects on Colonization Success in Mixtures of Gut Microbial Communities. Species introductions can play a major role in community assembly and change, but predicting their outcomes is difficult due to the diversity of species, community, and environmental properties that interact to influence colonization success. Colonization plays a particularly important role during the assembly of the human gut microbiome during infancy, and it continues to influence microbiome composition throughout the life of the host. But determinants of colonization success in this environment are not yet fully understood. Propagule pressure, which is defined as the size of introduced populations and the frequency of their introduction, is one major factor that can affect the outcome of species introductions, but its importance varies across environments, and propagule pressure alone may not be enough to predict colonization success in all contexts. For instance, in a clinical trial of microbiome therapeutics, propagule size, which was measured by inoculation dose, had mixed success on the colonization of the probiotic species Bifidobacterium and Lactobacillus. Several ecological factors can modulate the effect of propagule pressure on colonization, including the competitive interactions that often arise between introduced and resident species. Strong competition by resident species can sometimes exclude even large propagules, while weak or absent competition may promote the establishment of small propagules. Disentangling the influences of propagule pressure and competition on colonization success is crucial for predicting the outcomes of species introductions in the gut microbiome. In this project, I mixed and co-cultured a set of stool-derived in vitro communities of gut microbes at different volume ratios ranging from 1,000 to 1 to 1 to 1,000. To quantitatively assess the impact of inoculation dose on colonization success compared to other factors like competition, I compared the results of this mixture experiment to a set of predictions based on ecological neutral theory, called here the dose dependence model, which assumes that all species occupy the same niches and compete with equal fitness, leading to a regime in which propagule pressure is the only determinant of colonization success. Under this model, the relative abundance of every species in the mixture should be predictable based on the mixture ratio and the composition of each parent community. Alternatively, if inoculation dose plays a minor role or no role in colonization success, which I'm calling here the competition model, I anticipated that the relative abundance of each species in the community should be independent of the mixture ratio. Under the dose dependence model, I first predicted that community diversity should be higher in mixtures with a more even ratio of parent communities, as the relative abundances of more species, which we measured as amplicon sequence variants or ASVs, remained above a 10 to the minus 3 limit of detection. Instead, I found that the ASV richness of mixture communities remained similar to or lower than the level of parent communities across mixture ratios, implying that the diversity in parent communities before mixture may already be close to an ecological limit, and suggesting that introduced ASVs may need to outcompete and displace other ASVs to persist in the community mixtures. Although community diversity was unaffected by mixture ratio, I hypothesized that inoculation dose might still play a role in determining colonization success by influencing the abundance of ASVs present in the mixture. To quantify the impact of inoculation dose on mixture community composition, I used Jensen-Shannon divergence, or JSD, to calculate the similarity between mixture and parent communities at different mixture ratios. Under the neutral dose dependence model, mixture community composition should be predictable based on the mixture ratio and the composition of both parent communities. In actual mixtures, however, we found that community composition deviated from neutral predictions at every mixture ratio, and that for most combinations of parent communities, community composition remained relatively consistent across mixture ratios, suggesting that inoculation dose has a limited effect on community composition. I next sought to understand how community level phenomena like diversity and community composition might be driven by ASV level behaviors. Among individual ASVs in each mixture, I identified a variety of colonization patterns, including several cases of dose dependence, in which the relative abundance of an ASV varied substantially across mixture ratios. I also observed sets of ASVs that maintained consistently higher or lower relative abundances across mixture ratios than the neutral predictions, which I classified as overcolonizing or undercolonizing respectively. Notably, ASVs present in multiple mixtures often exhibited different patterns of colonization across different mixtures. For instance, one ASV from the Bacteroideaceae family, here shown as Bacteroideaceae 9, exhibited dose-dependent and undercolonizing behaviors in two different mixtures. Dose-dependent ASVs were present in every mixture set and accounted for a greater number of ASVs and greater proportion of parent community relative abundance than either overcolonizing or undercolonizing ASVs. These results suggest that inoculation dose plays a measurable but limited role in influencing the outcome of each community mixture. Overall, these results show that although community diversity and composition are not determined primarily by inoculation dose during community mixtures, Dose dependence is present among individual species in these mixtures and may drive overall community level signatures of dose dependence. The distribution of colonization behaviors, including dose dependence, is dependent on community composition and may be affected by species and community interactions, including competition.